You can see my presentation. There is no
Yes.
any problem, right?
Yes, we can
Yes,
watch.
teacher.
Fine. Okay. So my chat for today, I choose the one of the hot topics in today. It's microRNAs, but also my investigation is about immunometabolism. So there is an important role in this matter. So I'm gonna take this for the sharing. In the first place, I want to talk about obesity. Oh my, our class is fall down. Sorry. Wait a minute. Okay. So obesity is the prototype of immune-metabolic disease. It is considered a major factor that triggers metabolic risk and the development of secondary chronic illness like insulin resistance. Obviously, another pathologies can appear like metabolic syndrome. The susceptibility of a subject to develop obesity will depend on different factors such as the repertoire of the individual variations in an ensemble of relevant genes. No, their history of exposure to environmental risk factors and the interaction between the lifestyle and metabolism, which is also modulated by the gene regulators. Meanwhile, obesity presents many subclinical manifestations characterized by alterations in lipid and carbohydrates metabolism. At different levels, obviously, most of these changes is due to a low grade systemic chronic inflammation that favors the development of insulin resistance, and the adipose tissue is the primary anatomical site where insulin resistance disease takes place. In early stage, this tissue became inflamed. Insulin resistance is the first step to develop diabetes mellitus type 2. Novel findings of the immune response in the regulatory process and metabolic mechanisms may open new avenues in the treatment of the common complex diseases and well as inflammatory components. Research on basic and clinical advances in immunometabolism has evolved rapidly during the past years, like 10, when we don't know a lot of these things, and 5, well known, that the role and the detection and characterization of regulation of inflammation metabolic diseases with the comorbidity can play an imperative role. Nevertheless, the precise mechanisms mediating these relationships remains poorly understood. The interplay in regulation of inflammation and metabolic risk factors are complex. Okay, the inflammatory condition associated with adipose tissue represents triggering factor in the etiology of the obesity pathologic mechanisms and mainly contributes to the related disease outcomes. The identification of diverse molecular mechanisms related to energy metabolism has allowed the definition of strategies for searching genes implying in obesity and insulin resistance. So experimental reports show the existence of small non-coding RNAs, which are identified like microRNAs. the function of the regulating cellular processes through modulating the expression of genes that code for functionally proteins. Once synthesized, some microRNA can be released into circulation via exosomes, vesicular bodies, lipoproteins, simple extrusion, or by apoptotic cells. The most researchers in the field have assessed the presence of circulating microRNA in many body fluids, being related to the impaired expression in tissues under physiological and pathological conditions. Several studies have shown a correlation of particular circulating microRNAs with the development of different pathologies, positioning them as a valuable biomarkers in silent diseases such as obesity, insulin resistance, or any component of the metabolic syndrome. So the molecular biosynthesis process of microRNAs involving multiple pathways. However, it is possible to characterize a general mechanism in which sequential routes of a particular method I'm gonna tell you. In first place, microRNAs are transcribed in the cell nucleus by the RNA polymerase II, based on three main gene sequences: intronic regions, polycistronic clusters, or from intergenic areas. The molecules obtaining are called pre-miRNAs. The second step is these miRNAs are improved by RNase type 3. It's called DROSHA, to become a pre-miRNA, which is recognized by the exportin complex and transported to the cytoplasm through a nuclear pores. The pre-miRNA, once it reaches in the cell cytoplasm, dicer, it's another enzyme, cleaves the double-stranded fragment and release the loop. The miRNA duplex is unrolled and loaded into the complex called miRs. Once it is loaded into the RISC complex, the major miRNA is capable of associating with the mRNA target. Subsequently, when the major miRNA is formed, their function will be to recognize it by the 3' UTR complementary sequences in the target RNA messenger. So the level of coincidence in these sequences determines the degree of regulation of transcription, with one of two options. Okay, when the sequences of the complex is 100% complementary to the sequences of the region and of the target, I mean, perfect complementary, this leads to the denaturation of and degradation of the RNA messenger RNA, while incomplete complementary triggers silencing the messenger RNA through different molecular mechanisms also, as repression of translation, degradation, and it could be both, sequestration of the target. Since its deregulation has been related to different illness, and it is estimated that is an important data. It is estimated that more than 60% of the human genes expression are regulated by microRNAs. So I'm gonna talk right now about the diversity of these microRNAs only in the immuno-metabolism pathologies. Okay, I mean, the study of microRNAs, it is relevant because it has been involved in diverse functions and their expression levels have been associated with different diseases. So, I mean, insulin resistance, diabetes, atherosclerosis, or cancer. New microRNA sequences is identify their their participation in biological processes at the molecular level is to be defined. The mechanisms also, like adiposity differentiation, metabolic integration, and another one, appetite regulation. Among the metabolic diseases associated with expression of microRNAs, the role in diabetes mellitus type 2 has been described. I mean, where it was shown that one example is MIR 375. This is directly involved in the regulation of insulin resistance, insulin secretion, insulin secretion. And this suggests their participation in the metabolic pathways and the possible association with inflammatory markers and their expression in individuals with phenotypic characteristics associated with obesity inflammation. On the other hand, the miRNAs can also be found in a soluble form. I mean, the circulating form is the same thing, soluble, circulating form. And it is important to notice in this that they remain stable in serum for long periods of times. And principal for two mechanisms. One is the formation of a complex or ribonucleoprotein with argonaute proteins and the addition to an exosome particle. Besides, this is super interesting. It has been discovered about the association with high density lipoproteins that can transport mRNAs in the plants and take them to the target cells where they will be captured. So this is very interesting. Related to their location in the serum of the miRNAs in a mature form, many authors have proposed two hypotheses. First, the microRNAs of tissues can be present in the circulation as a result of cell death, no, and lysis. But how I was saying before that there is another way, that is the tissue cells can actively secrete mRNAs in their microenvironment with an exosome. With an exosome, they get into the blood vessels so they can get their way up the circulation. So that this has been also suggested their potential using as a markers for different diseases. And in a long time or period of time, we are thinking about using this like a therapy, like a gene regulatory therapy. Well, due to the strong correlation between the expression patterns of mRNAs and the state of the disease that has been shown with not only in animal models, I mean, in also humans patients, the mRNAs has been considered as promising candidates for not only biomarkers, like I say, therapy. So in immunometabolism, particularly, it is important to recognize that metabolic diseases have a strong immune component derived from inflammation and oxidative stress, in which microRNAs can regulate different ways. In diabetes mellitus type 2 association of MIR-146, MIR, sorry for the Spanish, MIR-126, this, yeah, that is more easy. With markers of endoplasmic reticulum stress and inflammation, as well as decreased MIR-20B, 21, 24, 58, is the 126, 191, 197, 223, 320. This is also like a control, by the way, and 408, 6. I mean, there is a lot of microRNAs described, okay? In obesity, there is many others, no? I mean, 162, the increase, 17, 138, decrease. I mean, this is microRNAs can increase and decrease in the period of time, in depending of the if we're talking about obesity, insulin resistance, diabetes, or any kind, or in the stage of metabolic syndrome. That is what I mean. Well, with regard to metabolic functions in the skeletal muscle and the regulation of microRNA species, it is known that can lead to deep alterations of glucose and lipid metabolism in adipose tissue, which is an important metabolic regulator. Regarding this, it has been shown that the expression of many microRNAs can decrease the levels of triacylglycerol and diacylglycerol. So this could be an important lipid regulator in this level. Also, there are other microRNAs that have shown that they affect hepatic insulin sensitivity. And more recently, is shown that the increase of some of these microRNAs improves glucose tolerance and insulin actions. That is why we are thinking that use them like a therapy, like a gene regulatory therapy. So I don't wanna bore you guys, sorry, but I'm almost. Oh, okay. I got the idea, sorry. So the association between obesity and fatty acids, different microRNAs have been associated, okay? Among them, there is 2 of them that I love it, that is MIR33, the family of MIR33, A and B. They are found in the intronic regions of the genes SREBF2 and SREBF1 that go for the transcriptional factor that they have the control of the synthesis of cholesterol and fatty acids. So this family of microRNAs have a lot of my interest in, because they act as suppressing genes that has the opposed the functions of these transcription factors like SREBP2 and SRBP1. So for instance, the cholesterol flux and the oxidation of fatty acids So that under low cholesterol conditions, the transcription of SREBP2 and the regulation of genes involved in the synthesis of cholesterol and absorption is activated. Therefore, co-transcription of the miRNA 3T, the family 3TA and 3TB acts in exporting cell cholesterol, inhibiting the transcription of the ABCA1. So, the higher levels of free fatty acids and different adipokines such leptin and resistin have a strong regulator role in other microRNAs, while the expression of MIR143 can modulate the differentiation of pre-adipocytes by increasing the storing of lipids. Also, these microRNAs blocks the differentiations of adipocytes through the synase 5 regulator of extracellular signals. So, in resume, I don't know if I can explain myself, but I want to tell you guys that this is a new world of words, and these molecular functions have an important role in these days. This is a hot hot topic in science because they can regulate the expression of the messenger RNAs. So this can regulate the expression in the level of the protein. So we know it, and we can manage it in another level, in a therapy. Probably I'm gonna check up my handles for use the models, the experiments, for use this like a therapy. So thank you for the attention. I don't know if you have any comments, any questions. I'm ready for hear you guys.
Thank you, Fer. Thank you so much. There is one question. I know it's been answered in the chat. Fernanda asked, what is the meaning of miRNA? And Melanie also answers microRNA, but I don't know if you can tell us, for the people who doesn't know too much about molecular biology, what, in the simple word, what is microRNAs? What is it?
It's an RNA that is a small RNA that no code. This is no coding for a protein. Okay, there are many types of RNAs: messenger, ribosomal, transcriptional. So microRNA is a small, no coding RNA.
Okay, Melanie has a very good questions. I don't know if you can open your chat. And
yes, yes. I'm seeing the question of Melanie.
Melanie, do you want to open your microphone, or it's okay if we read it? What do you prefer? You can. Do you want to open your microphone, Melanie? I guess it's a no.
That is a no.
It's a no. Okay.
I know my baby. She don't want to talk.
Really, please. Okay, so you can answer her question.
Melanie says that when you talk about using microRNAs as a form of therapy, you mean by inhibiting or activating them. And how could this be possible? By creating microRNAs in vitro or by using some sort of competitive molecular to inhibit the function? No, I mean that use this in case that are a form to be synthetic. I mean synthesize a molecule, a change, a small change, and this is gonna be called, because exist, and sirna, or there is another one that is mRNAs. So you can use this synthesized molecular. I mean, this is not natural. This is making a laboratory. And then I have the idea, I don't know. then use this in an appropriated vehicle. This is the problem, the vehicle to transport to a cell target. So the proposal, the dream is use this to regulate the expression. I mean, many of the pathologies, the problem is that there is one protein or two proteins or many proteins, inflammatory proteins or anti-inflammatory proteins. I mean, one phenotype or the other phenotype, no, that it's dysregulated. And I mean, one example. I don't like this disease, but I know about this disease because this is the life. It's called rheumatoid arthritis. You know about this disease? The principal mechanism of this disease is inflammation. Is the chronic inflammation is present every day in the patient. So if you can use RNA that you are not blocking, you are not knocking, you are not knock out a gene. No. You are regulated the expression. That is the dream. Can regulate the expression if we can use one of these molecules in one. Once again, we need a vehicle. That this is the grade with only watch it. So you need a vehicle available to export these molecules, and they have the potential to trigger the cell, the I don't know how to say it, the target cell, no, the important to regulate, I mean the chondrocyte. So where is the articulation? Where is the inflammation in the articulation? So that is the thing. So this is a dream. Obviously, it's a dream. I hope that this dream could be true in no less 10 years.
Thank
I
you.
don't know if I can explain.
Yeah, I don't know, Melanie. So I have another question related to the question of Melanie. So these microRNAs that you talk about, they are synthesized, is right now in this sale. You can buy it. It's only for research. You can buy it for.
It's only for research. Only for research, and it's a lot of expensive. It's not easy. You can buy it. You can buy it, but you can buy the sequence that is described. That is a natural sequence. You can buy it, but you can buy it, but you can't use it because there is no vehicle. This is it still remains in experimental. I mean, models in vitro in animals, but it's still in protocols and investigation because someone is missing. Something is missing. Is the vehicle that allows that this molecule resist.
Okay, so you think, I don't know how, but these synthesized microRNAs, a good vehicle
they call siRNAs or SN or RNAs. They have two names. It's different, but I mean that is the thing.
It can be used the nanotechnology for these microRNAs. Do you know that? Yes. Okay.
I have thinking about that. I have dreaming about that. I have 4 years thinking in that, thinking and write it and thinking and how I'm gonna do it, because it's not only a dream. I'm sure that we can do it. I'm pretty sure. So it only takes a lot of money for investigation, please.
I know that there are a lot of students that are in the medicine, right? So I would like to know if these microRNAs as a biomarkers can be used in the clinical. I mean, obviously. Can you do with a patient
Yes, yes, and it's easy because you only need a blood sample, and only that. It's
And,
like it's not invasive. I mean, it's
but there is some laboratories that can, you know,
Mexico, no.
no,
Mexico, no.
no. Okay,
In
so
Mexico, no.
it can be really handled with a patient. That was my question. I don't know if any guys has a question. You can open your microphone. Don't get nervous. We can help you, and or you can write it down so Dr. Fernanda can answer all your questions. And remember, this is a webinar. You can express and the language as the way you can learn it. So you learn writing, listening, and also with the different topics of medicine, no? So anyone has a question? Anyone wants to ask for something? Or you can write it down. No, please don't be shy.
There are colleagues I want to thank for attending this webinar, and also, sorry, I want to thank a lot of my students. I love you guys. Thank you for attending this. And also, there is I'm seeing the people that are seeing this, and their colleagues also, professors. So I'm very grateful for this. Thank you. And sorry because um, I talk a little quickly, and so I'm sorry if I cannot explain myself.
No, it's okay. So I would like to remind you that I'm gonna open my computer again. So I would like to remind you that there are going to be 5 different webinars every Tuesday at 7 p.m. We are going to start this Tuesday. So I hope you can join us. The webinars can last between 30 minutes to 60 minutes. So it depends of the professors that can join us. So next Tuesday, we're going to talk about respiratory disease in the neonate. So I know that some topics are really related to medicine, but if you are a nutritionist, a public health, or gerontology, you can also join us and learn a lot and make some intervention of it. Remember, these webinars are for learning and practicing our English. So, and also I would like to remind you, if you open your chat, you're gonna see a link in Google Forms. That link of Google Forms is for, you can get your certificate attendance if you join us with 5 of the 6 webinars. So please remember, it's gonna be open the first 30 minutes of each webinar is gonna be open, this link, so you can write all your data, and you can get that at the end of the webinars. So these chats have been really interesting. We really appreciate what you did, Fernanda, because whatever, will you believe it or not all professors can be doing this. I mean, you can invite the professor, but not everybody accepts. So we are really thankful for you and for your assistance and for all the talk that we have done here. So Fernanda, we appreciate it. And I hope you can see you the next semester. And I hope you can join us the next webinar and the next Tuesday. Also, tomorrow is going to be another webinar, but that webinar is going to be in Spanish. So
everyone to the Spanish webinar. It's going to be more easy.
Yeah, it's going to be more easy. It's about molecular biology in the immune system. It's going to be by the Dr. David de la Mora. Yeah. And it's going to be in Spanish, so you can join us, guys. And if you can, the level of the protein, so we know it. When you can, when it's kind of funny, the noise of your computer, I don't know, you can repeat the question or the thing that you were saying. You can repeat it, please. You can say it again, please, Wendy.
Wendy, sweetie.
Why not? You can ask him. He was invited to give one webinar in English, but maybe next semester, maybe he will.
Maybe next semester he can he could do it. I don't know. I don't know why, but
Not
we
easy.
don't know why. But maybe next semester, maybe.
Yeah. So Wendy, I don't know, we lose you. So you can write the question down or you can write us. So if there is not more questions, I would like to finish with this. And thanks, Fernanda, again for joining us and joining everybody. So
Thanks everybody. Thank you a lot, and thanks for being comprehensive. And oh, also my glass of water. Hold on. I don't know if you noticed.
no,
I have a glass before my computer, so my cat always, my loving cat. Uh, my glass with full of water. So I like. Oh, I'm sorry for that. Okay.
thank you so much, guys. See you next Thursday or see you tomorrow with David de la Mora. Thank you so much, everybody. See you next time. Bye.
Bye.
Thank you. Bye.
See you. Thanks, teacher.
See you.